Hi everybody. Thank you very much for coming, as they say, and I'm very glad to have the opportunity to speak, and thank you very much to the organizers for inviting me. I've moved recently, or over the last 2 or 3 years, into this concept of translational medicine, and while preparing this talk, I realized I haven't really ever thought about defining it. And it was quite worrying, actually, because I thought, I've been working on something that I can't even define. And so I had to think about that, and then I started to read about it, and it's quite tough to define something where you have translational medicine institutes, and it's quite exciting to see that the field, I speak to other people, and they pretty much agree it's a difficult thing. Just before I get going, I just wanted to point out that I do consult for a company, and that a number of the slides today are influenced by Russ Altman's top 10 translational bioinformatics that he does every year, where he reads out and finds awesome biomedical informatics translational papers. And that's good because some of them, two of them coincided with ideas I had, so I felt good. But at the other point, he's been a great influence on my work, and I just wanted everybody to know that he's a great guy to follow. So translational medicine, well, basically it's scientific discovery and health improvement. That's a no-brainer. But then when you look under the hood, there are many, many tracks through that. And there's also a strong component of public health intervention that doesn't often come across in translational medicine as a classic field. And so thinking about that, when you start to think about what underlies clinical change, clinical implementation, we tend to think about it, as you know, from a clinical record perspective or interacting with clinicians and giving them stuff. When in actual fact, there's an enormous space of the way that people work and the description of the clinical environment done by the clinician and within their work. They're very intuitive people. And we tend to think about the delivery that we have to clinical as being somewhat quantitative, somewhat organized. And really, when you think about it, it's very much dealing with people, bringing quantitative information to people. And that's a classic of what we do as computational biologists and bioinformaticians. So if you look at the path history, the roller coaster of translational medicine and drug discovery, it's great because it's basically a bunch of failures with the odd success, just enough success to keep the investors punting and giving us more money. And effectively, we start off in the 50s with pharmacology and biochemistry, physiological approaches, closely allied to academics. Then we move away a bit from academics into the 70s with affinity assays and the disparate relationship of drug companies and rational drug design differing from then the roller coaster hits the beginning of the biotech period and into the 80s. I don't know about you, but monoclonal antibodies were the thing when I was a graduate student. And effectively there, we have the speculation and the rapid growth of the concept of investment into biotech. And off we go then through a bit of a dip as it crashes, and off we go into the area where we are now, which is the mother of all tsunamis. And effectively there, we're looking at the individual genome projects becoming many genome projects, and the associated data coming from that, not only from quantitative assessment of genome variation, but other aspects of data which are coming and agglomerating with that and being shared and attached. And effectively, that is bringing on a whole new paradigm. And that really is where we are right now, and it's quite exciting. And then, of course, we've got to store and work with this, and we've heard about some of the infrastructural issues. And there's many to describe, but the basic issue of this all is that it still takes a very firm hand on the tiller to make progress in a field that is buffeted by so many different opportunities, directions, and basically discoveries. So, a long time ago, actually, in '93, I went to a meeting and Lee Hood said that we are doing biomedicine, it is a data science. And at that time, he said at least 20 to 22% of what we're going to do is going to have something to do with data. And I think that's changed a bit. But in other words, large-scale data biology. And I think what's exciting about that is that we've moved to a point now where we see what's happening at the Institute for Systems Biology and other groups. And it's amazing to think that we've actually progressed in a very short amount of time to a point at which we're really taking data-driven answers quite seriously. And certainly during the rise of the ESTs in the early days, I remember being at ISMB in '96, and I was one of the few people at that meeting that didn't go for a job interview at SmithKline, and they just hired up a huge chunk of the field, and we knew who was hiring was being interviewed because they would have a toothbrush in their pocket after their interview. So it was very fun to laugh about it. But effectively, I think it's quite important to realize that the companies very quickly realized that it's going to become a data issue early on. So now we know that clinicians are learning to type. And I was very excited to hear that I'd be accepted for a job in UK and I could move there and I could work with the National Health Service. And that's when I discovered that most of the record taking with general practitioners was by hand. They were using the odd computer for fun. I was quite amazed. And I've seen, interestingly, we've seen quite the dynamic range of record taking also in South Africa, where I'm from. And it was intriguing to me that really the advent of IT has still got some teething issues in terms of being widely adopted in the medical system. So it was really amazing. And I think that's changing, but it's a very crusty environment to change. And really what works best is to start from scratch. And in terms of moving to having a very IT-driven interaction within the hospital in a real-time environment, that's still a long way away, certainly in the United Kingdom. And I see some innovations of that in the States, but also I note that really it's driven there very much by individual companies seeking to reduce costs. So the ability to take on a particular pipeline or perform a particular assessment is driven basically by the capacity of the individuals within the system. And when it's balkanized, it's very difficult to see that people will be embracing this whole world that we're so excited about. So the word of caution is that to deliver on the ideas and the work that we do here, it's still quite some ways from the battlefront. And even when you are working with people at the battlefront, you notice that really it's an issue of training. And that also is a very long-term investment. Talking of which, this is a very old slide, but effectively we're looking at this issue of population sequencing. And the first whole genome sequence of a population published significantly has been the Icelandic population. The Faroes Islands, 50,000 people, 100,000 in UK, 300,000 Qatar, all slated to be done, 1 million slated to be done in the States. The issues of scale that come with that are challenging, to put it mildly, but the reality is that with that data, when and if it becomes available, I think the integration of that with patient data is going to be very exciting indeed. And that's one of the major reasons that I wanted to come actually to work in the UK, because it was institutionalized in the description of what's occurring. And I was excited by that. And I really do believe that we have a great opportunity, and we have an opportunity to change things. And the work that we do within this society is going to fundamentally change the way that we do modern medicine. And when I say that to modern medical people, they don't look at me as if they believe me, and I can't think why. But I do believe that what we're up to is going to be very transformative. If you take a look at diagnostics, the first direct-to-consumer wave has hit. And I took a punt on this because my sister has a severe disease, and my family, we took a look. And of course, I found out, yes, she has genes for brown hair, but it wasn't terribly informative. And actually, that isn't unusual. And Kara Pikoff, as a journalist at the New York Times, also took a look at this problem, and so, as did many others. And effectively, there's plenty of opportunities to go to companies for direct-to-consumer information. One of the issues, though, that's quickly been arisen there in the commercial area is that the evidence behind predictions is very, very spotty. And Clara's work showed that basically going to different companies could give you different values for the same disease, based basically upon the biases in the background of the tests. But what's exciting and interesting about that is that the idea is there. The implementation, I think, is still quite a long way off. The core problem is a lack of evidence. So actioning on clerical variation is a lot of what we do, and I think the field is very much centered around that, triaging variants. And this is an important paper that came out with Dan MacArthur, but effectively the process of doing this is still very nascent, and that's why this paper is important. And I've noticed that because I've actually sat down and worked with clinical scientists in the hospital and asked them, okay, so exactly what is it that you're doing? And what the generic words that are coming back are, well, we sat down and we did this and we did that, and we put the script together and we got it approved and we did this and we did that. And that's very much a disappointing response, because really, as you know, we're all looking for an aspect of clinical validation that's very much shared. So it's exactly the same as in any technology. We haven't flipped yet into a place where there are standards that are broadly applied. So there really is a challenge in clinical diagnostic interpretation. This is a model I took from the website of Sophia Genetics, but effectively I'm using it to show how things are occurring in the UK. And you heard a lot from Tim Hubbard, but effectively the Genome England Genome Medicine centers are interacting with the clinical partnerships, which are supposed to be analytical groups that are working with them. Now, none of that really exists yet, but it exists in name. But the exciting part of that is that the local councils are getting together with this. And the politicians actually are investing here more than they're investing here, because the local councils are really into the idea of smart health cities. And I'm seeing that amongst my colleagues in the UK. And the combination of that is really where things, I think, are going to happen. And the paradigm shift that represents has been recognized by Eric Schadt at Mount Sinai, and he's taken on a very serious approach towards building the new hospital, the data-centric hospital, the data as the patient hospital, where intervention and wellness is up front and treatment is what you do if those are not successful, moving information to the patient and interacting with the patient in the home. So the clinical enlightenment that comes from learning from this is a long way off, but I think certainly we have to work to help it happen. So in terms of therapy, this is another very exciting area. And having worked for a while in the Stem Cell Institute, it's a very, very rewarding area to be working in terms of seeing how people think. The paradigm of going to veterinary or animal model trialing and working with animal models is a very expensive one for drug companies, because it doesn't work very often. The translation between systems doesn't work very well at all. And moving from preclinical research to human clinical trials actually is a paradigm that is being picked up quite actively, because it's turning out to be cheaper to do this and more effective, obviously, to do this than this. And I'm excited by that, because that's where technologies that we can produce and help with are becoming very important, particularly in the area of transdifferentiation and building stem cell models of diseases. And one of the first papers in that by Egan and Wilf in ALS, an area that I work, really demonstrates that it's possible at least to recapitulate a model and to potentially ameliorate the symptoms. But the problem with this, of course, is that you're looking at a model and the symptoms that are demonstrated by a patient's transdifferentiated fibroblasts are not going to be the same. Excuse me, the symptoms, the manifestation in a cell, this group of cells in a dish, is not going to be the same as in the patient. And that issue, the phenotype recognition and the connectivity to the phenotype, is a very, very big one. And so the adoption by industry is very slow, actually, in this area, considering the potential for performing experiments in human lines. Another work in the regenerative area is very exciting. Doug Melton has children with type 1 diabetes, and his work at the Institute has generated these stem cell transdifferentiated cell models, which really do work very well in terms of ameliorating hyperglycemia in mice. But really what that does is provide a very stable and testable platform for therapeutic development, disease modeling that is scalable, and it seems to be adopted more readily by companies, which is interesting to me, being that it's made in a different species. So the key paradigm that is being adopted, I think, with some interest, is the shift from single gene directed work to multiple gene interaction networks in terms of targeting. And that's important. And it's important because, as you know, even these exciting studies that occur in cancer that work on single gene targeting don't work that well, because the systems become refractory to that very rapidly. And coming up with the correct combination of drugs to address that is almost an NP-complete problem. So understanding with the networks of interaction of genes is fundamental to, I think, taking any form of causal-driven drug therapeutic intervention forward. And building that upon that is something we'll hear more about from Anna later today. I'm very much looking forward to her talk, but effectively bringing information together and fusing it into single networks so that you can improve and improve and improve on modules in terms of their p-value association with the phenotype. And this is work, and the concept of this has been driven hard by Eric Schatz's group, where he published a paper on TyrBP as a very important molecule in the process of late onset Alzheimer's. And that group now that he's part of, and that I'm fortunate to be working with, the Accelerated Medicines partnership is really exciting, because what's happened is that there's been a drive from the funders to say, we want you to work together. We want you to do things better, and we'll give you the money. There's been a drive from industry saying, we want to reduce risk because we don't have the capacity to interpret the data that we're generating or that we're trying to work with. We need to find trusted partners. And the partnership is inside a trusted walled garden where effectively it's a preclinical consortium of academics and industry. Now, that I always turn off when I hear that. I don't know about you, because it's like, it's another huge, great big thing. We'll see a result in 10 years, maybe. But this is real. And what's exciting is I've attended these meetings for 18 months. I'm very much into understanding the social interaction. And what's exciting about it is that the group that's working, and they're very good, these are very leading scientists, very, very strong-willed industrial partners. They trust each other and they're working together very closely. And the only thing that's happening, not quite yet, is the combining of the models that people are generating. And that requires some form of sharing platform. And they've adopted the Sage Commons platform, Stephen Friend's Sage Commons platform. Other interesting things is that companies within the consortium, such as Takeda, have said, we'll put out all of the data that we find in terms of validating the models that you provide us with. We'll share it publicly. We won't tell you perhaps what drug we're using, but we'll show you how things are working. And I think that's fascinating because it allows us to understand that there are aspects of the drug industry that will change because they desperately need to reduce risk because of the huge fallout rate of drug candidates. So the last part of this talk, how am I doing for time? You want to know? 4 minutes? Great. The last part of this talk is just on this issue of pathways and networks. And I'm interested in one thing that I think has value, which is why I'm taking the time to explain it to you. Otherwise, I would continue on the broader issue. But effectively, if you can take a look at a set of interacting genes, and you can look at the way they interact with a background data set, and you ask that background data set, say, geocorpus, how do these genes interact across the geocorpus? And you define the boundaries of the gene set. You can ask then about the correlation of the internal correlation of a set of genes. You can call it a pathway if you want, but most canonical pathways are not highly internally correlated. But you can build an interesting thing. You can build a relationship between sets of genes in a canonical context across this background. And that allows you to ask the question, how do all of the known gene sets, say, in pathway space interact? And those which interact positively, you can have a positive correlation. Those which interact negatively have a negative correlation. And that allows you to say that when this pathway is up, this pathway is down, and vice versa. And that sounds very simple, but if you think about it, it's actually quite valuable, because what it allows you to do is paint a functional experiment into a space, and then to attach information to that functional experiment based upon other criteria that you have. And that allows you to be predictive without having to know every single element of every edge and interaction in a fixed network. It also, okay, so we are working hard on this. We're actually clustering these pathway sets and just to see what's happening. It's not published yet. So one of the things I thought I'd show you is that you can do this with the Go biological process, for instance. And if you sample on axonogenesis, you can show things which are associated with, but it's different than enrichment. What you're actually showing is the relationship between these processes that are labeled. And we have a statistic which allows us to compensate for any gene overlap. And that means that what we're looking at is the influential relationship between these processes. Yet another way to organize Go, yes, but what it also allows us to show is interesting things, such as the fact that mitosis is slightly down-regulated potentially in axonogenesis. And that may or not be true, but I think what's exciting is that it gives you a relationship that you can query and help you understand the mode of action of a drug. So if you want to look at the relationship with drug response profiles, you can go ahead and do that. We've got some exciting new work coming out soon in that area, and we can see that we can predict certain drugs that may have certain actions by just looking at the data. Now, everybody does that, but what's useful, I think, is if you know how right you are. That's a challenge, particularly in drug repurposing. And anyone who has some smart responses, smart ways to assess how right you can be, I really love to talk to, which is why I put up this slide. So there are plenty of roadblocks and challenges. I thought I'd take something ethnic, as we say in South Africa, with respect to Ireland. And we do some discussion here, a little bit about data sharing and provenance. A recent commentary in Nature, driven by Lincoln Stein, puts forward the issue that we have very large data, and it's getting much larger. This is an issue I think that Ewan's given us, and it's not new. But effectively, we really do need to think about this cloud environment. And programming and working in the cloud is not as easy as it sounds. Certainly, moving a lot of our code to the cloud has not been trivial. But what's interesting about that is it gives you a chance to share your genomes in the same space. And I really am excited by the idea that if we can share data sets in a really confined, in a well-structured environment, there's a really good opportunity for us to start doing a new form of data federation, which, as you know, in this field is a non-trivial challenge. So in terms of provenance, I wanted to mention Sage Commons, which has a system for publishing workflows as nano-publications with DOIs. It's very useful if you're teaching people how to do things, because you can look back. It's also very useful if you want to gain some recognition, say in a GitHub environment, for what you're doing. Also within the Stem Cell Commons, which is built upon a similar model that we've built at the Harvard Stem Cell Institute, we have put in a basis by which you can easily submit your data set as a data publication. And we'll keep the workflows next to the data so that you can reproduce the data. Here's just a classic sort of website, but what I want to bring to the point here is that you can add code to the system as well as browse it. And I think it's important, new paradigm for working with data systems, not just to have them static. Finally, I wanted to mention that we're trying to encapsulate the concepts I've given to you today at the Center for Genome Translation, which is a newly created center at Sheffield Institute for Translational Neuroscience, where I'm fortunate to work. And there, at the University of Sheffield, there's this view that faculty and scientists should collaborate and work closely together, and they do, which is kind of why I went there. And what's important to bear in mind is that the domains that we cover are very disparate. And so you have to try and work across these domains, and one of the best ways to do that is to share students across those domains, which is what we're doing. And what we care about is looking at this target prediction and repurposing. But what we need to do in order to do that is to move from genes to networks and processes. We have to have a very good handle on phenotypes. We have to have a very good handle on what the patients are up to. And our models need to be very, very reproducible, and they need to actually be predictive. And those are all challenges all of us face, but I wanted to bring it into one space for you to look at briefly. So in terms of Future Watch, I think the concept of real-time trials is coming. Very close, very quick. And there's a very exciting trial that SmithKline is working on, where they're taking fibroblasts and strands, differentiating them and treating the fibroblasts, and also looking at how the patients respond to particular drugs and taking molecular profiling from that. The aspect of going from treating patients to predicting and preventing issues in patients is a big one, particularly in the States, where it's been driven financially. It's being picked up also, as I mentioned, in the UK in a different way in terms of being the right thing to do. It's very hard because you have to bring together many communities to do that. The aspect that I haven't mentioned a lot of today is the machine learning approach has been taken by companies such as IBM, where they're trying to incorporate as much knowledge as possible into a machine learned environment so that they can advise doctors in the clinical record. And that's a very exciting concept when you think about it, based upon the fact that the doctors who are needing to be advised via looking at a clinical record are not capable of reading everything which is out there. Finally, I'd like to mention two aspects. One is that we're going to really, I think, have a much better handle on the relationship between genetics and environment, because we're counting the environment a lot more. I mean, a lot of you, I'm sure, are wearing aspects that are counting your environment right now. And I'm excited by the concept of face space, which is the prediction of how you look from a few key marker SNPs. And artists have taken that up first. Forensics is following closely. And I'm really looking forward to the social networks starting to ask you for genome data. So thank you very much for entertaining this talk. I hope I haven't been too content-free. We are hiring. I'm allowed to say that once, I hope. Very much looking for postdocs and research associates. And I entertain any questions.
We have time for one question. If you have any burning question, please come to the mic.